Assalamualaikum and hi, my name is Husna Hafizah binti Fazli with the student ID 2020483216 and my supervisor is Dr. Wan Nurhayati binti Wan Hanafi. Welcome to my presentation to my panel and all the viewers. So today, I'm going to present about my final year project that is Comparative Analysis of DNA Extraction Efficiency from Strawberry, Blueberry and Bilimbi using a DIY method. So now, we will start with the first part, which is an introduction. All living things in this world that are made up from the cells will have the DNA, and this DNA can be obtained by DNA extraction. Scientists have made incredible strides in developing extraction techniques. DNA extraction is a technique to separate DNA from cell membranes, proteins, and other biological components from a sample using physical or chemical processes. The DNA extraction that will be used in this research is a DIY method. This DIY extraction method is the simple method which only uses home living substances such as onion, detergent, salt, and others. In this study, different kinds of fruit like strawberry, blueberry, and bilimbi were used for DNA extraction. They have the same component which contains the phenolic compound that includes the phenolic acid, flavonoids, and polysaccharide. The problem statement that inspires me in this project is there is insufficient research on DIY methods to identify the suitability of this method to be used on different fruits, and there is no study about the concentration and quality obtained from this extraction method. Then, we will move to the second part, which is objective. There are two objectives for this project. The first one is to compare the DNA extraction efficiency among strawberry, blueberry, and bilimbi, and the second one is to determine the quality of extracted DNA from strawberry, blueberry, and bilimbi. The third part is methodology. In order to accomplish the first objective, the method used was sample preparation, DNA extraction using the DIY method that was adopted from University of Utah 2008, and gel electrophoresis. From the sample preparation, juice of fruits will be obtained, and from the DIY extraction method that originally done on strawberries, it will be applied to blueberries and bilimbi. So, the formation of the clump in each sample will be observed. Then, to accomplish the second objective, the spectrophotometer was done to obtain the result of purity together with the concentration. Now, moving on to the crucial part, result and discussion. The first result is obtained from the DIY extraction method, which there are DNA clump formed on strawberry, blueberry, and bilimbi sample. Clump intensity shows that this method is efficient on strawberries compared to blueberry and bilimbi. However, Table 1 shows that even moderate intensity of clump produces high concentration reading on spectrophotometers. The gel electrophoresis shows that sample BL1 and B1 produces the brighter bands, and according to Stewart 2023, larger DNA concentration result in brighter bands. However, the size of the band falls between 1250 base pair and 1000 base pair. Therefore, no DNA concentration reading on spectrophotometers obtained on blueberry and bilimbi samples means that it was possible that the DNA in the sample extraction was too little, or there was no DNA existence in the sample extraction. So, the spectrophotometer cannot detect the existence of the DNA in the sample. Next, to obtain better result, an optimization was done. After the optimization, the overall results shows that improvement in concentration of DNA. There were readings on fruit which previously had no reading, especially on bilimbi. However, the results for strawberries has become slightly lower than before. The optimization that was done was mashing the fruits and using freshly picked fruit. By mashing the fruit, only break down the cell wall, while blending the fruit, the blade possibly damage the DNA. Damaged DNA can cause genomic instability, apoptosis, or senescence as stated by Hakim 2008. Then, freshly picked bilimbi is better than non-fresh bilimbi, and it was supported by Yamamoto et al. 2006. Fresh fruit contains higher DNA than non-fresh fruit. So, it shows that by optimizing the steps and substances, the DNA extraction can be efficient on other fruits like blueberry and bilimbi. The third result is obtained in order to achieve the second objective, which is to determine the quality of extracted DNA. Table 3 shows that there was no purity reading obtained on sample strawberry 1, blueberry 3, bilimbi 2, and bilimbi 3 for original results. This happens because the concentration of the sample are close to the lowest detection limit, which is lower than 10 nanogram per microliter. The result that was not in the range shows that it was less than 1.8, which means the sample was contaminated with protein. The contamination by residual guanidine, phenol, and other reagents used in the isolation protocol also caused the purity not in the range. Carbohydrates are the common contaminant related to nucleic acid isolation from plant sources. However, after an optimization was done, the reading of purity becomes better, and the optimization done was by replacing cold water with cold distilled water. Cold distilled water reduces the contamination of the result. Distillation removes all contaminants from water and eliminates 99.9% of the minerals that are dissolved in water, as stated by Blades 2019. Overall, we can see that the quality of the strawberry DNA is the best among other fruits. Now, for the last part is conclusion and recommendation. Conclusion, it shows that the DIY extraction method was efficient to use on strawberry, and a little optimization makes this method efficient on blueberry and bilimbi. Then the greatest quality of DNA was obtained from strawberry samples. However, bilimbi also shows the potential of getting good DNA quality. My recommendation is to further the study by amplifying the extracted DNA that had been obtained from a DIY extraction method using the PCR. It is to show that there is an existence of DNA that can be used in further study. Here are the list of the references that were used in this project. That's all from me. Thank you.